This is episode 216 of the Stem Cell Podcast, Liver Tissue Development and Engineering, with Dr. David Hay. Hello, everyone. We are Doctors Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. We are very excited to announce that not only will Arun and I be at the upcoming ISSCR annual meeting taking place in San Francisco from June 15th to the 18th, but the entire Stem Cell Podcast team will be there as well. That's right. If you're attending the meeting, drop by the exhibitor hall to find the Stem Cell Podcast booth and learn how you can be featured on a future episode. We really can't wait, have been looking forward to this for like 2+ years. Today, we have Dr. David Hay from the University of Edinburgh on the podcast to talk about his research finding novel ways to produce liver tissue from pluripotent stem cells. We've also got our usual roundup of recent highlights in stem cell news coming right up.
But first, the Stem Selfie contest is back. Enter your best cell image by April 28th for a chance to win a Stem Selfie prize package, which includes a magnetic puzzle of your image. Visit www.stemcell.com/stemselfie, that's STEMCELLFIE 2022, to find out more about the contest and to enter. And don't forget that voting starts on May 2nd.
Wow, that is really big news. The Stem Selfie contest is back. I love the Stem Selfie contest, but I got bigger news than that, Arun. This is a bombshell and so fundamental to all of our work in the stem cell field. We're talking about chemical reprogramming, okay? You know, reprogramming and the foundational study, seminal work by Yamanaka, I mean, it really kicked open the door for pluripotent stem cell research and therapeutic application. But ultimately, you've always had to use cellular factors. I mean, transcription factors in the most case. Early days, it was delivered by virus, and then more methods that were more amenable to translation were developed. But nevertheless, we've also always had to deliver transcription factors or like ooplasm or something in order to affect that reprogramming. Although, you know, it was almost a decade ago now when work led by Hong-Q Deng, who is at Peking University in China, showed that you could use chemical reprogramming, that you could use chemical stimulation in mouse somatic cells to generate pluripotent stem cells with an elaborate protocol and a cocktail and all that. But nevertheless, they could do it. And it was a big deal. And this approach was extended even to direct lineage conversion by Deng's group and others, as well as Sheng Deng at UCSD. But none of these approaches were able to be translated to induce human pluripotent stem cells from somatic cells. And I think a lot of people attributed that to the fact that human somatic cells have evolved to be have a more stable epigenetic landscape. So they were more resistant, so to speak. But the idea it was still there, and in the mouse they showed that it could work. So it was reasoned that there was perhaps a more plastic state that existed in other animals or in other cell lines under different conditions. And so Deng's group here hypothesized that they could recreate that plastic state in human cells and affect that chemical reprogramming. And this is work. It's no wonder that it's taken almost a decade since this work was first shown in mouse before it was translated to human, because this must have been a monumental amount of work. And it shows in this manuscript, which has 8 co-first authors and 3 corresponding authors. And what they were able to do was create this intermediate plastic state in human cells. First, they did it with fetal fibroblasts, but ultimately were able to do it with adult dermal fibroblasts as well. And by creating this intermediate plastic state, notably by inhibition of the junk pathway, they were able to create cells that exhibited key features of embryonic stem cells. And that was, you know, transcriptomic analysis, they looked like pluripotent cells. The epigenetic bisulfite sequencing, they were a match. Teratomas that generated all 3 germ layers. They went on to do single cell RNA sequencing during the reprogramming process to kind of map it. And ultimately, I thought this was the real kicker. is that they then mechanistically kind of unpacked it and showed using axolotl, frog, and mouse that this intermediate plastic state looked more like the axolotl program following limb injury than what you saw in frog and mouse limb injury. And axolotl famously is able to regenerate limbs. So this was really, I thought, the capstone of this study was showing that bit of mechanism, showing that it was akin to this, I quote unquote, lower, although I would say more glorious organism that's capable of regeneration. And that's the kicker here. I mean, not just laying the foundation for generating pluripotent stem cells with a chemical cocktail from adult human dermal cells, which, sky's the limit there. But I think maybe even planting the seed that chemical reprogramming could be modulated at some level to affect a kind of regenerative process like what we see in axolotl. And that really blew my mind here. I think we've been so focused for all this time about chemical reprogramming to get pluripotent stem cells. But now reading this, I'm envisioning a future where you just have refined chemical cocktails that allow you to de-differentiate to a more plastic state and then directly differentiate towards specialized tissues, cells, perhaps someday tissues and even organs. So I think it's a really exciting piece of work that's been a long time coming. And I would say next to, Yamanaka and induced pluripotency, this and similar work done by Deng over the last decade is such a critical addition to the tool set that we have.
Yeah, this is a tremendous story. And really, all the stories that we're going to cover on this week's roundup are really top notch. This is one of those roundups where you want to really just listen to every single story very carefully because these are very high profile papers that we're covering today. But in this particular story, yes, I mean, this is a tremendous amount of work over many years of basic science, many years of screening to find the right cocktail of compounds that would be able to induce chemical reprogramming in human cells. Certainly, there's a huge amount of high throughput screening that was done here. Just looking at the methodology and looking at the cocktail that they actually ultimately used, it's complex. This was definitely not an easy thing to find. It's not like you're just looking at one single small molecule that's going to induce the process of reprogramming. This actually kind of reminds me of our chat with Elias Sengach, who's also doing kind of similar work when it comes to finding cocktails of small molecules that can enhance cell survival in their lab's case. This is even more complex than the cocktail that they described, because for reprogramming, as we know, it's a multi-step process. I mean, if you look at the methods in this particular paper, you have to prepare media for 4 different stages of induction, right? And each of those stages has a slightly different cocktail of small molecules that you're using. It's a huge amount of work here. I will say one thing though, small molecules, of course, as we know during our differentiations that we use them in, they're cheap and they're effective in a lot of situations, but they can be somewhat dirty, right? So they have some of these off-target impacts, off-target effects. I don't know if they characterize those in depth, right? This is certainly non-integrating when it comes to the genome, and you're not using any sort of viral mediated reprogramming here, but small molecules will have their own caveats too, right?
Yeah, that's a great point. I mean, both your points there, which is that the complexity of this, and I would say I'm kind of heartened to see that, or it's a bit of a relief, right? Because you would imagine that if it were a simple cocktail, that you would have this plasticity or de-differentiation happening at a higher level, maybe in endogenous tissues, leading to some kind of cancers or what have you. So I'm not surprised, and that it would take a lot to override this program. Cells don't want to drift once they're set. But on the other point, I would say it's also true that the idea of just applying these, I wouldn't even see willy-nilly, but even applying them in a very targeted way, there's probably more than a few unknown unknowns in terms of what kind of off-target influence that compound would have, even in the cells you're targeting, notwithstanding the result that you want, that de-differentiation entrance into plasticity and perhaps directed to another fate. there may also be other effects that you would worry about short or long term. So something that we have to really pay close attention to. But I'm sure you would agree, Arun, just a breakthrough conceptually, technically, and the fruit of a lot of labor. So I really appreciate the efforts by all the scientists and those who came before making this come to fruition.
Absolutely tremendous, tremendous work, and really a labor of love in a lot of ways over many years of painstaking screening and all that good stuff, right? So hopefully the next step is being able to adopt these cocktails, this technology for everyday reprogramming. We'll see what happens. Moving on to another really high profile paper that's a bit different. This is a basic science study, but it's also a tremendously high profile study in Nature. The title of this one is Somatic Mutation Rates Scale with Lifespan Across Mammals. And this is mostly an aging related story. There is a stem cell element to it that I'll get to. This is coming from the Wellcome Trust Sanger Institute. Last author here is Inigo Martin Corena, and there's quite a few authors on this paper, but first author is Alex Kagan. This is a beautiful basic science study. It's a comparative evolutionary biology story. It's really the first study to compare mutations across a bunch of different animal species that's answering a really basic fundamental question about aging and cancer, right? The In a nutshell, why aren't bigger animals, say like the elephant, or animals that are able to live longer, why don't they necessarily have a higher propensity to develop cancer? This is something that's puzzled a lot of folks over the years. In fact, this is the basic fundamental principle of what's called Peto's paradox, which I actually didn't know about prior to this. It's a paradox where bigger animals or animals that live longer don't necessarily have a higher propensity to develop cancer. Intuitively, it would make sense if they were more inclined to develop cancer, right? If there's more cells and there's more time for those cells to accumulate mutations, then perhaps there's a higher chance of those animals developing cancer, the ones that are older or bigger, but that's just not the case, right? So they're trying to get to the bottom of this. And what they did is they actually analyzed genomes from different species of mammals, all sorts of mammals, from mice to giraffes, small ones, big ones. And they confirmed that really the longer the lifespan of the species, the slower the rate at which the mutations occur. So it's a really cool discovery. So it doesn't matter how old the animal ultimately becomes on average, the number of somatic mutations that are accumulated over that lifespan is the same. It's just the rate of the mutation occurring in the animal that's changing. So for example, if there's an animal that only lives a very short amount of time, like a mouse, its overall mutational rate, somatic mutation rate, is much higher than an animal such as like a human that lives for 80 plus years. Overall, our mutation accumulation rate is slower. And they're able to basically figure this out using, and here's the stem cell angle, using intestinal crypts. Okay, so basically they compare the intestinal crypts from different animals, different mammals. They're able to measure the mutation rates in single intestinal stem cells, right? It doesn't, the thing that it doesn't answer, this particular story, it doesn't answer the question about body mass. Why is it that larger animals are not necessarily inclined to develop higher amounts of cancer? That's something that this study doesn't answer, but it does help to answer that the lifespan question. Okay, so really a beautiful basic science study, a lot of sequencing here, a lot of fundamental biology, a lot of evolutionary biology, comparative biology to answer this really fundamental question in aging.
Yeah, it's so like touched emotionally when I read a study like this, because it just reminds me the beauty of nature and how these harmonious equilibria just arise out of the system. It's lifespan. Now add this to the catalog. Lifespan and sexual reproduction and all these factors that have to be balanced to maintain a species. they all work in concert. And this, it's interesting to see now this kind of genetic or mutational angle. The other thing that I'm wondering about this is, as you said, it doesn't address the question of body mass, but for me, it also, what comes to mind is the mechanism, right? It's nice to see that these relationships work, but how do the different species mediate that differential mutational rate? And I know in elephants, at least, because, you know, the most massive, the most famously studied, it's known that they have 20 copies of P53. So there you can kind of surmise, yeah, maybe that's how elephants have evolutionarily been able to modulate their mutational rate. But I'd be really interested to see similar analyses of all the, you know, DNA repair and tumor suppressor genes, how they are represented across the gamut of all these species, just so we could get a peek at maybe how mechanistically the gene expression is able to mediate this kind of balance that you see.
Yeah, absolutely. I think it's great that you brought up the elephant point. I mean, that's probably the most famous example of how a large animal that's able to live for a long time perhaps is able to protect itself from developing cancer phenotypes. You know, the 20 copies of P53 in the case of the elephant, a number of which are actually active, by the way. But then there's other animals, other species that are also have these unique mechanisms. There's the naked mole rat, really famously, which is also I don't believe it's able to develop cancer and it's able to live a significant amount of time. So I think there are, it perhaps is not as clear cut as you might think. Maybe different species have independently evolved different mechanisms to protect themselves against different cancer phenotypes. And then the other part of this is, you know, this is focusing just on mammalian species, right? So it'd be really cool to see in plants or invertebrates or whatnot, you know, what sort of unique mechanisms do those species and those organisms have to protect themselves against cancer? It's just a beautiful reflection on the diversity of the evolutionary tree, don't you think?
Absolutely. I love diversity. Speaking of large and small, all animals, large and small, you know, they have a lot of variation, but really it all comes down to one cell, Arun. It's one cell to rule them all. And that is the hematopoietic stem cell, because no matter your size, just one cell can create your entire hematopoietic system. That's in mammals, at least. I don't know about the plants. You're wandering into plants, Arun. I want you to come back to me. We're talking about mammals here. But, you know, hematopoiesis, as glorious as it is, and the potential of one cell to generate the entire hematopoietic tree, uh, perhaps owing to that tremendous capacity, it's poorly understood how it works out. We know that developmental hematopoiesis consists of multiple waves, and ultimately culminates in the generation of these self-renewing hematopoietic stem cells that you find in the bone marrow. And we know that these cells emerge, at least in humans, in the aorta gonad mesonephros region. And although that region contains numerous hemogenic cells, these endothelial precursors of hematopoietic cells, a transplantable hematopoietic stem cell, something that will repopulate, is very rare. There's about one, it's been calculated, a single one of these powerful cells per AGM, okay? And that suggests that somewhere in between their migration from the AGM to other sites, they undergo a maturation process that creates the capacity for this engraftment and differentiation potential. Where they go from the AGM is varied. They go to the liver, and there they have the fetal liver. There they acquire this ability to engraft the bone marrow, and then ultimately they go to the bone marrow. But we haven't been able to recapitulate all stages of this in vitro, or been able to observe them all in vivo. And although the establishment of hematopoietic stem cells is known in the fetal liver, there's multiple progenitor waves that lead up to that. First in the yolk sac, then in the fetal liver, ultimately, as I said, in the bone marrow. But there's all these other sites too. You know, yolk sac, placenta, even in major arteries, in the head, in the heart, Arun, there's a site of hematopoietic stem cell dissemination. So the contribution of all these sites to the establishment and development of hematopoietic stem cells is not known, particularly in humans, because it's not an accessible model. right? And the key here, it's thought, and one major reason why we aren't able to track these is because we don't have good labels. We don't have the ability to distinguish between bona fide established hematopoietic stem cells and their immediate progenitors, okay? So enter Hannah McCullough, who's a real goddess of the hematopoietic stem cell field and has made a lot of seminal contributions. Also, many collaborators on this as well that I won't mention, but have a look at the paper. This is a real dream team who unpacked the progression of hemogenic endothelium to transplantable hematopoietic stem cells, also visualize this using spatial transcriptomics. And what they ultimately found is that there's this hematopoietic stem cell signature, and I'm going to say it because it's really important. RUNX1 positive, HOXA9 positive, MLLT3 positive, MECOM positive, HLF positive, SPINK2 positive. Half of those things no one ever has thought about in the hematopoietic field. So this is a really novel marker that distinguishes hematopoietic stem cells from their progenitors throughout gestation. And they also showed the transition of these stem cells to the liver is marked by a molecular shift where they take on these more canonical maturation markers, including CD133 and HLA-DR. And they also tracked the origin of the hematopoietic cells to these specific hemogenic endothelium. ALDH1A1 positive, K17 positive hemogenic endothelial cell, and then showed that those cells came from a precursor that was an arterial endothelial subset identified by IL-33 and ALDH1A1 positive. Finally, they go into a pluripotent stem cell system and generate what they call correlates of these cell types. Although they do acknowledge that there are some discrepancies between the pluripotent stem cell derived putative hemogenic precursor and derivatives. But nevertheless, I mean, I cannot overstate how important a study this is, because we've identified McCullough's group and her colleagues, they've identified these landmarks, these milestones that are clear phenotypic indicators of all the steps along the path. And I think now it's just a matter of trying to fish out these cells and generate mouse reporters, et cetera, so that we can really observe and experiment with them. And that's what I'm waiting to see here. All the transplantation studies that should follow from this, both in the mouse system, as well as taking these human cells, maybe from aborted fetal tissue, and seeing how they'll repopulate NSG or immunocompromised mice. So there's so many studies that I can't wait to see coming out of this. I just want to get into a pluripotent stem cell system right now and mess around with some of these markers and see if I can fish out a pluripotent or a hematopoietic precursor, because, I suffered under the yoke of my efforts to make hematopoietic cells, and I would like to give it another shot. That's my great dream, Arun.
When I told you about this paper, I saw you salivating about it. You're just so excited to cover this one. And really, this roundup is next level, man. It's probably top 3 roundups I've ever done while I've been on this podcast in the last couple of years. Every single paper we're covering here is just big, big news. And this is no exception. I mean, you can just think about the applications for the discoveries that they're finding here. And the thing I love about this is this helps explain perhaps why we're not able to generate those bona fide HSCs, hematopoietic stem cells, from our pluripotent populations. And it has to do, according to this paper, with the niche, right? If these precursors are traveling all over the body to be specified correctly, then perhaps we're not recapitulating that in the right way in our dish, right? But that's the cool thing. This is perhaps providing that roadmap as to how we can improve our HSC production in vitro as well. So I guess I'll turn it around to you and ask you a question, since you're the HSC guru on this particular show. Why did it take until now for this to come out? Is it because of the technology? Is it because of the samples that are required for a study of this caliber? Why is this coming out now? What do you think?
Well, yes, I think you said it right there. It's limitations on the availability of tissue, and also, yeah, we've arrived at, I think, the culmination of our ability to examine heterogeneity within populations with single cell sequencing. And on that note, I will say that there's still a bit of a ways to go. They did use, and I mentioned, I think they use spatial transcriptomics to localize this. I don't know if you've worked with the spatial. I've done something with that with the 10x chromium Visium system. And it's amazing. I mean, don't get me wrong, the applications, but in their work as well as my own experience, I note that the resolution of the beads, it's a 50 micron area that you can capture with each bead or cell, as you would call it. And 50 microns for me, looking at the ovary, encompasses an entire group. It entires a primordial follicle and all the associated cells. So we're not exactly at the level of single cell resolution with spatial. And I think similarly in this study, there's still some challenges in being able to resolve single hematopoietic cells within a 50 micron region. So there's a little bit of a generalization that they need to do in their gene expression. But I can't wait for the next generation of these spatial transcriptomics technologies to arise, because for sure they're going to move down that resolution to 10 or sub 10 micron beads. And I think that that's going to give us an even greater edge in all of our studies. But in this case, it would allow us to really localize and define that niche, and move this even further. And the short answer to your question, Arun, I think less than the availability of tissue, it's the tech, man. The tech just drives this stuff forward. We see things, every iteration of new tech, we have to look at everything all over again.
Yeah, I think that's probably the right way to answer that. I think, yeah, like you alluded to, that's the other cool thing about this is because it seems like it can get even better, right? If your resolution can go down to the single cell level when it comes to these spatial transcriptomic studies, then perhaps we can figure out even more unique markers and pathways that can utilize to make our HSCs better in vitro, right? So I can't wait to talk to you about the next iteration of this story, which perhaps is going to come down in the next few months. Who knows? But we're going to wrap things up with another very high profile story in the roundup here today. This is a science paper, and this is also one of your favorite topics. Sorry about stealing your thunder here, Dalen. The title here is Functional Primordial Germ Cell-like Cells from Pluripotent Stem Cells in Rats. This is coming from a number of Japanese colleagues of yours, I'm sure. Last author here is Toshihiro Kobayashi. First author is Mami Oikawa. And also importantly on this paper is Hiro Nakauchi from Stanford, who is known for his work in chimera studies. This is a very big deal, as I'm sure you would agree with. This is basically the first time where researchers have been able to create healthy, fertile rat offspring from sperm that are derived from rat pluripotent stem cells. Okay. So it seems like in addition to the mouse, where this is already kind of possible, deriving, primordial germ cells, precursors of sperm and egg from ESCs, mouse ESCs, which is done like 10 years ago. Now it looks like the rat has joined that club. And this is it's a pretty simple study when it comes to at least the figures that are being presented here, but it's certainly a tremendous amount of work that's been going on in the last 10 years to get to this point. They're able to induce these, primordial germ cell like cells from rat pluripotent stem cells using a variety of different factors. And then, ultimately, I think the real beauty of the study is that these are functional. Okay, these are functional spermatids that are being derived from these rat pluripotent stem cells. They're able to do the knockout system, knock out, germ cell and gonadal development in rats, and then rescue them with some of these primordial germ cell like cells derived from the pluripotent stem cells from rats. And then those rats were able to have viable offspring. I think there was a caveat here that I don't think there was able to be natural mating with these particular animals. I think there still had to be some sort of assisted reproduction that happened to actually produce the offspring. But still, a landmark study, because in addition to mice, now we can create sperm from rat pluripotent stem cells. And ultimately, it's coming down the road. This is all working towards making it from human pluripotent stem cells. I think you're perhaps a little more pessimistic than I am when it comes to the reality of the situation, whether that's ever going to happen or anytime soon. I still think it is, but you're the expert here, right, Dylan?
I wouldn't say that. I'm just an enthusiast, and my takeaway from this is just what we need in the world. Another way to make more rats. Um, Riding the subway, I would disagree, but in all seriousness, this is I have been I wouldn't say outspoken, but I'm on the record saying that I don't think it'll ever be safe to make human children from in vitro cultured pluripotent stem cell derived eggs or sperm. But who cares what I think? This is amazing. And also, like, forget about humans for a second. It's really important. Even if I don't think we should do it in humans, that doesn't mean that we shouldn't figure out how we could do it in humans, right? To have the capacity, to have the tools, and putting that aside, just species preservation. I mean, the way we're going, we're blowing up all these animals to extinction. The white rhinos, northern white rhinos or southern, I can't remember the most recent example. And this is a real viable potential way to, I'm not talking Jurassic Park here, but make, newly extinct species, to maybe reinvigorate them, or to support the revitalization of some near extinct species or already extinct species even. So, I mean, putting aside my qualms about the application in humans, I have to uh, pay my respects and kudos to this group, because it is really a much more difficult process. The infrastructure isn't really there for mice. I mean, for rats, the way it is for mice. The reproductive biology is different. Our understanding of rat pluripotent stem cells and the tools we have to manipulate them are scant by comparison. So this is really a monumental piece of work, and I think we've reached a threshold now where we need to be encouraging researchers to be moving higher into monoovulatory species, and beyond. Well, I don't know what's beyond that, but just to try and expand this application to other species to see how far up the ladder we can go.
Yeah, that's a great point. I think the human side of this is perhaps something we don't even have to touch on because, like you said, there's a huge application here for preservation. And I remember we've been talking to Jean Loring, who was a guest on the show a while back. Remember, she was making those banks of iPSCs from different endangered species, like the white rhino, for example. And this is the next step of that, right? If we can make those banks of iPSCs from different endangered species, this kind of work will help us figure out actually how to make the sperm and egg from those species, which is ultimately, yes, going to lead to new offspring and new members of that species and lead to better conservation and preservation of that species. So it's a beautiful piece of work, and I'm sure there's going to be other species, other animals, other mammals that are going to uh, follow. And I think everything happens in the mouse first for a reason, right? But like you're alluding to, it's not always the best model of reproduction, and this is one step towards making another better model of studying human reproduction, right?
Yes, all comes down to the humans, and I have to laugh because every time a story like this comes out, all the attendings in my practice here, they get a ton of calls from patients saying, "Hey, they're doing it in rat now. Can you make sperm from my skin?" So then I have to field all that fracas. But very important piece of work and a monumental impact to come. So we'll look forward to that. Now to talk about the liver with Dr. David Hay. But before we get to that, a quick message from STEMCELL Technologies. Are you looking to advance your liver research with organoids? The Hepatocult Organoid Kit can help you easily and efficiently establish, maintain, expand, and differentiate human liver organoids for your experiments. Organoids grown using the Hepatocult Organoid Kit are suitable for studying hepatic development, metabolism, regeneration, and disease, and are suitable for studying drug metabolism and drug-associated hepatotoxicity. Visit us online to learn more about Hepatocult at stemcell.com/hepaticult, HEPATICULT. All right, everybody. Today on the show, we have a special guest from the UK, Dr. David Hay, who is chair of tissue engineering at the Center for Regenerative Medicine and the Institute for Regeneration and Repair at the University of Edinburgh, also founder of Stimuliver and STEMnovate. Dr. Hayes' group is defining novel ways to produce liver tissue from pluripotent stem cells. They use in vitro derived tissue to better model human liver physiology and to develop supportive cell-based therapies for disease. Dr. Hay, thank you so much for joining us today on the show.
Thanks, it's great to be here.
Well, it really is certainly great to have you. My introduction there was a bit cursory, a bit threadbare, especially considering both the tremendous scope of unmet medical need related to the liver, as well as your outstanding contribution to meeting that need. Why don't you start us off by briefly reviewing the devastating impact of liver disease globally and the major goals of your laboratory?
Yeah, so that's a great question. I mean, liver disease is a devastating disease. Over 2 million people die annually from diseases associated with the liver. And, the liver is a remarkable organ. It performs so many functions within the body. It has a high regenerative capacity. And those attributes are lost in the case of chronic disease, where you start to get healthy tissue replaced by scar tissue, which not only removes the functional cells from the liver, but also distorts the architecture, which creates this problem where you can have some patients which progress and other patients which don't. But the ones which do progress, have incidences of liver decompensation, where their organ doesn't work properly, which isn't consistent with normal health. And those can have other knock-on effects on other organs. But really, when you start getting to that end stage in the disease process, your treatment options are very limited. If you're too sick for a transplant, then your chances of survival decrease dramatically because there are no other treatment options for the disease. So it is devastating. It affects people's quality and quantity of life.
And what's the focus of your lab in trying to address that major unmet need?
So what we're trying to do is generate renewable sources of human liver tissue, really so we can understand disease in a dish. So try and better understand the disease and maybe develop new medicines to treat that and improve patient outcome, but also trying to develop supportive modalities that we could generate liver tissue which could be deployed within the body to either promote regeneration of the host organ or support the host organ until a transplant becomes available, or do both. And that's really our goal, is to try and give these patients that have no treatment option a treatment option.
So the workhorse cell type that you've been using to actually generate some of this replacement liver tissue has, of course, been pluripotent stem cells, and you've been focused on the production of human hepatocytes from pluripotent stem cells for seems like more than a decade now. And it seems like actually one of your biggest claims to fame was the development of this really efficient differentiation protocol to actually go from human embryonic stem cells to functional hepatic endoderm through modulation of Wnt signaling, published back in PNAS in 2008. And I'd like to think that differentiation approaches across the board, no matter kind of what cell type or tissue type you work in, have been streamlined over the last decade and a half, in part because more folks around the world are actually using human pluripotent stem cells in their own tissue specific research. And it's partly because of iPSCs, right? Induced pluripotent stem cells. Everybody started to use these pretty usable, accessible stem cell populations for their work. But when it comes to hepatocyte differentiation specifically, which is actually something we talked about with Matthew Sinton from your lab, get us up to speed on the current state of the art. How robust is the differentiation protocol for hepatocyte production from pluripotent stem cells? And how mature are these hepatocytes that we actually get at the end?
Yeah, that's very interesting. So there are a number of differentiation protocols out there which generate efficient levels of hepatocyte-like cells. They employ different, as you were saying, different signaling factors which promote the efficient differentiation and the maturation of these cell types in culture, and generates a product which has mixed abilities. I think it's fair to say that the cells display fetal and adult traits. Certainly in our hands, we're able to demonstrate the metabolism of drugs that you would see in the adult. So, for example, the cytochrome P450s that are involved in prescription drug metabolism are active in our system, but they're also active in our system at a point where the cells are secreting a fetoprotein, alpha-fetoprotein. So I think the cells have mixed identity in the dish. And we're currently working to try and understand that a little bit better. Is that mixed identity a cell-specific trait, or do you have different populations of cells in the dish? And part of the problem of working in 2 dimensions is the cell models that we create aren't long-lived enough to be able to mature substantially in the culture dish. And this is why we've turned to 3-dimensional differentiation and cell aggregation, really to try and move past this instability in the culture dish to improve the maturation of these cells and ultimately their application, whether it's in the lab or whether it's in the clinic.
Yeah, and that's the thing. I mean, we've made such great strides, and now we're dealing with kind of a whole different animal than we were considering when we first started working with pluripotent cells. And in the field, progress has been so rapid. Every episode, we highlight some novel translational study that affirms and/or underscores the tremendous clinical impact and potential of cell-based therapies. But I can remember, like Arun's alluding to there, back in 2008, you know, when faithfully recapitulating the differentiation of a single specialized cell type, in your case, the hepatocyte, was a huge deal. We couldn't get the kind of, I guess, coordinates, the molecular morphogen gradients correct, or we didn't have the recipe right to get exactly the right cell. But so it was big. But now we're working at different scales, different levels of complexity. You got to integrate multiple cell types in an organoid or assembloid to get a paper out. And we're constructing these biomimetic scaffolds to endow these in vitro kind of artifacts, I guess you could call them, with the structural elements that you see in biology. And I would venture, here's the question, I would venture that we generally underestimated how tough it would be to reach the projected therapeutic endpoints, right? I think we all, not all of us, myself, I can speak for, had this idea that let's get a cardiomyocyte, let's get a hepatocyte, and we can address heart disease, liver disease. But clearly, it's a lot more complicated than that. What do you think? And what are maybe some of the underappreciated hurdles that remain to be bridged before we can really get to that therapeutic endpoint?
Yeah, that's a great question. I think the issue with being able to produce these high quality cells has been probably the complexity of our differentiation approaches in the past. Before, we were using matrigel, we were using feeder layers, we were using fetal bovine serum, and all of these are susceptible to batch variation and introduce complexities in your cell culture system that makes it very difficult to get that reproducibility in the process. And that was one of the first things that we tried to do, working with defined signaling molecules, using simple commercially available medias, reducing those to the minimum amount of levels that gave us the ability to produce our hepatocyte-like cells at about 90% purity, which was the real game changer for us. Before that, we'd been in the, 0 to 20% yields in our differentiation process. And, the work done by Kevin Demure, looking at activin A, certainly with the endoderm, was, really important for us in the field to be able to make that, highly pure population of endoderm that we could then further specify down the hepatocyte lineage. I think now we've got to that point, and we've worked with industry to be able to produce stem cell differentiation media that allows us to flick the switches consistently in different cell types with different users at different densities. So I think we understand the kind of differentiation landscape much better now for hepatocyte differentiation. I think going forward, there's a limit to what we can do in 2 dimensions working with one cell type. I think we need to consider the other cell types that are in the organ that we're interested in studying. And certainly with us in the liver, the incorporation of endothelial cells and stellate cells has been really important to generate a kind of phenotypically stable piece of liver tissue that has the capacity not only to perform metabolically in the dish, but also to be able to model aspects of disease, whether that be fatty liver disease, or whether that be cancer metastasis. These are both really exciting avenues that we're exploring further now with our kind of defined pieces of liver tissue. And one of the key things that's got us to this point now, I suppose, is in the incorporation of automation too. And that really tells you how good your differentiation process is. If you if it's reproducible between users and you're satisfied with it, and it then is suitable for use in a liquid handler and automatic pipetting systems that allow you to scale up, then that's fantastic. I think you've really nailed it. Then we can use that high throughput system to understand what we have and what we don't have in the dish in a little bit more detail, and then start refining our approaches to differentiating the cells, but always with that goal of having something that's chemically defined and something that's simple for the end user to perform, to get the piece of liver tissue that they want to perform their experiments with or go clinical with.
Yeah, I think reproducible differentiation is the foundation for everything we do. It doesn't matter what tissue type or cell type you work in. I'm a cardiac biologist, so reproducible cardiac differentiation is really important to me. And hepatocyte biology is the same way, and I think having these chemically defined approaches has been really critical. It allows for multiple people across the field to actually jump in, and also it gives us an eye towards the clinical and GMP grade manufacturing, which is what we're hoping to get towards down the road. And now that we've actually been able to make cell types like hepatocytes reproducibly and in a chemically defined fashion, we can really have more of an eye towards the applications, which is the exciting part, right? So when it comes to utilizing pluripotent stem cell derived hepatocytes, whether it's 2D differentiation or 3D differentiation based approaches that you use to actually make these things, it seems like there's three big applications. And I think this is kind of the case for a lot of different tissue types as well. I think cardiac is the same way, disease modeling, drug screening, and clinical transplantation, right? And certainly there's been a lot of progress that's been made on each of those applications, modeling different types of liver disorders, like what you were talking about, identifying potentially hepatotoxic drugs in preclinical settings. This is a big problem with drug development, and certainly using stem cell derived hepatocytes for transplantation purposes and for clinical therapeutic purposes to actually restore liver function. I mean, in your opinion, and this is probably going to be a tough question, but in your opinion, probably what's been the most promising of these applications? Is it the preclinical side of things, the identification of toxic drugs using these these stem cell derived hepatocytes? Is it the disease modeling that's been really promising, or is it really the clinical facing applications of using these cells for therapeutic applications? What have you been the most excited about?
I think initially we were, we, like the rest of the field, were very excited about this potential to combine our stem cell based models with genome editing and disease mutations to try and understand and cure disease better in the dish and try and take that towards the clinic. And I think that's really exciting. I think there's so much more mileage in the in vitro modeling side of things. Where I think we're kind of limited by our imagination now, right? We've got to be able to do something that's scientific and worthwhile, but I think we've got many tools at our disposal. More recently, the clinical side has really excited me. And I think, there have been a number of nice papers out there where people have been able to transplant cells and support liver function in compromised recipients, and that's great. I think, what got us really fired up was this ability to be able to put a bit of liver tissue underneath the skin and support the host organ from a remote site. And, maybe for some patients for patients who are very sick, this might be their only treatment option. They may not survive general anesthesia. At that point in their body, the blood's probably not going into the liver, it's probably shunting around the liver, which means that you probably wouldn't want to put liver tissue into that diseased organ because it's not going to be properly perfused and the cells are going to die in response to the necrotic environment. So I think, to sum up, I think definitely the in vitro side is really exciting. There's going to be a lot of good stuff coming. This is a really solid pipeline. But the in vivo side now is starting to develop, and that's probably been our focus in the last few years, is to try and get these implantable modalities to treat failing liver function.
Yeah, I like the way you put that. The in vitro side has gotten a lot of mileage. We've gotten a lot of mileage because I don't want to call it low-hanging fruit because the work is tremendously complex, but we've been able to glean some really important insights already clinically using pluripotent stem cells, induced pluripotent stem cells, and CRISPR and all these things. So it's already reaped some rewards. But as we all know, the holy grail here is clinical treatment using cell-based therapies. It's what we promised to the public with all these, public-facing funding mechanisms. And you alluded to it earlier. I think one of the really important elements to bring that into fruition, and that you've worked on, presumably in service to your overarching goals, is automation, right? The incorporation of automation into diffs for therapeutic application. It's a no-brainer. But my question to you is, how deep do you expect this to go? I mean, we just talked a couple episodes ago about this deterministic, or I don't know what you want to call it, AI adjacent method for cell selection and sorting. And the idea there is that the machine is looking at the cells and deciding which ones based on whatever criteria, in this case, morphological criteria. And AI is already being applied in my space, reproductive medicine, for embryo tracking and selection. So this is already in the clinical space. And in the case of pluripotent cells, these cell differentiations, it can be nuanced and often dependent on, I mean, you alluded to earlier about how there were all these complexities with the FBS and the matrigel, and you've made tremendous efforts to cleaning up that system, but still there's some kind of nuance and vagaries of differentiation, and sometimes a effective diff is dependent on, or at least can benefit from, the judgment of a committed trainee overseeing them, or PI, whoever it is. So this is the question. I mean, when it comes to automation, how far do you think we're going to go with this? Are we talking like HAL level IA, from 2001? Are we talking about humans that are running relatively dumb machines?
Good question. So I think the likes of AI, machine learning, are very important. As long as you've got the data sets to train and produce the correct algorithm that's going to be able to, streamline your process. I mean, my dream is to go in on a Monday, push the green button, and then come back 2 weeks later, and I've got liver spheres at the other end, that allows me to go and do the exciting science and maybe not a lot of the laborious work. I see AI could be a very important part of that setup. For me, it would all be about streamlining the process, but in terms of making it simpler, right? Getting rid of additives that are not required. What are the minimal components we need to make a piece of liver tissue? That works very well for the in vitro side of things, but also for the in vivo side. When you start working with regulators and physicians, they want to know, they want the paper trail that goes with that piece of liver tissue to, not only justify its quality, but also its suitability and safety for long-term deployment within the body. So I see this as a very important part. I'm no expert in AI or machine learning, but it would be a component of my ideal system if I could design that robot to be able to produce liver tissue.
Yeah, that regulatory point is actually, I think, a really good one. And it's not something that we think about a lot, especially when it comes to GMP grade production of cells for cell therapy. It'd be really great for the the regulatory agencies to be able to track your cell products, and having an automation based approach, it really allows you to perhaps track them a little bit more efficiently, as opposed to having different technicians who may have slightly different approaches in terms of making their cells. There's more variability when humans are put into the picture, right? So perhaps from a regulatory standpoint, that's attractive to automate all these cell production based approaches. And really, I mean, like what you're talking about, the end game is the clinical translation, and this is something that you're really excited about, like what you've alluded to. And so we actually wanted to bring up a company that you've actually founded and are the CEO of. This is Stimuliver. It's where you're taking basic stem cell derived hepatocytes, and stem cell derived hepatocyte cultures, and you're implanting them into people to perhaps treat clinically failing liver function. And this is, of course, in human trials. And that's the downstream hope, is to put these in people. For Stimuliver's approach, you're actually taking organoids, right? You're taking these 3D liver organoids and placing them under the skin, like what you're alluding to help restore patients' reduced liver function in cases of liver failure. So tell us actually more about this startup and where the company is right now in terms of this timeline to ultimately bring these clinical therapies to people.
Yeah, so the work was initially done in the laboratory 2018. We were able to prove the concept that we could put liver tissue, so cells underneath the skin, and support the host organ in an animal model of tyrosinemia type 1, so in a disease situation. Since then, we applied to the Bioinnovation Institute's Venture Lab program, and we were funded to start up Stimuliver. And our goal really is to take this procedure that we've developed, produce hepatocyte-like cells, produce endothelial cells, allow them to self-aggregate in microwells, scale out that process initially, and then add those liver spheres to implantable devices. So in our case, it would be a non-woven polycaprolactone fiber. So allow those cells, those spheres to attach to the surface of that, then put them underneath the skin and study the supportive value of these spheres. And also their safety long-term is paramount. So where we are in the process now is the company's established. We have our RAs working on the differentiation, and we just secured a grant actually last week from the Danish Innovation Fund to be able to automate the process within the company as we move forward to our preclinical safety testing. And the goal really is to create high quality liver tissue that we want to study its safety profile and also its supportive profile. Going forward, we will obviously, as we've discussed, want to be going into the clinic if this is safe and appropriate to do so, targeting acute on chronic liver failure initially. Trying to provide those patients with an option where other treatment options don't currently exist.
I mean, this is so great. I love doing this show. I mean, I exist in the world, so I would benefit from all these therapies anyway. But being here, just having run the arc of talking about them like science fiction, and then listening to you tell me that you're funded, you're really on the cusp of making it happen. It's just so exciting. I mean, it's an exciting time, not just for everybody who could benefit from the treatment, but it's really exciting for the stem cell biologists, especially those who are part of the efforts to bring cells into the clinic. But all of us, I think it's really tremendously gratifying to see that this choice we made and this commitment we made to the field is actually bearing fruit. But bringing it actually to the clinic is a gargantuan effort. And some might say it lacks the romance, I guess, of the small working group sharing this aha moment at the microscope or looking at a novel data set. I mean, it's a bit of a slog, right? So is it tough to move between these two worlds for you? I mean, you kind of need a different mindset. And here's I mean, do you have to cultivate a different sort of leadership in that setting? And do you think there's different types of scientists or trainees or RAs, as you call them, that thrive in that setting relative to a more basic research setting? What do you think about those?
Yeah, I agree. They're two different mindsets. One's more kind of lateral thought process, and the other one's very focused on developing this defined product, and you're developing your target product profile and then moving that towards a clinical trial, securing the investment to be able to do that, interacting with regulators. I mean, one thing that's been, I think, is very important as you develop this commercially is to build structure within the company, and you get people who want to work in those particular areas. That's their interest. And I think, the RAs we've got are really fired up at the moment. They see this as an important challenge to treat liver disease and take forward the work we've done in the lab and try and produce something which is going to impact positively on human health. That's been great. I mean, for me, I like both environments. I like the lateral thought process. I like to be able to find new things out. I like to go through data sets, crunch data, have debate about things. And then the true acid test is, if you want to translate that, is the science good enough? And I think that's what we've really focused on recently is, reproducing what we've done in the lab in different pairs of hands, using different cell lines. And yeah, we're all really fired up about taking this to the next level, which is a preclinical validated approach that the regulators like and allow us to move into clinical trials.
You're living the dream. And it kind of seems like you're representing a new breed of translational stem cell biologists, because in addition to being an amazing basic scientist and having over 15 years of pluripotent stem cell biology experience, It seems like you've always had this eye towards bringing these technologies towards the clinic and developing products, right, through startup companies. Stimuliver is one example. You also started and worked on other companies, such as STEMNovate, which is focusing more on drug screening, I believe. So tell us a little bit more about this passion about founding biotech startup companies, how you actually manage to balance your academic commitments. Seems like you're a pretty busy guy. And also, maybe if you could wrap things up by telling us some advice and giving us some advice for the new generation of stem cell entrepreneurs. I'm sure a number of whom are actually listening to the show who might want to follow in your footsteps.
Well, I think it's been great to work with great people. So Rishi Sharma, who's the CEO of STEMNovate, and Dagmara, who's the CSO of Stimuliver. It's working with the right people with a shared vision, and they work equally as hard as I do on these different projects. And I think we make a great team. Teamwork's everything. I mean, regenerative medicine, if you're creating an in vitro model or an in vivo system that you want to or treat people in vivo, it's about combining these different what would you say, different characters, different capabilities, different disciplines, and then trying to mix that all together in a kind of defined recipe to produce something at the end which is useful, whether that be a microfluidic device that allows you to predict liver biology in response to drugs or new treatments or to viruses, or create new diagnostics, or whether you want to create this implantable piece of liver tissue or maybe other tissues, for example, going forward, tissues for the heart, like a bandage you could use, or encapsulating cells that you can put in other parts of the body to treat certain deficiency states. I think it's all about team building. It's having the right people on board that share a common vision. And this work is hard. As you said, this isn't like a walk in the park. This is a good number of years of team effort. And I think we all gain a lot of experience as we go through this. Nothing is ever the finished article. It's like when you publish a paper in science, you finish that paper with a full stop, but that's just the pause before the next paper comes on. And I think that's pretty much in the corporate world. It's exactly the same. It's about developing the process, hitting your milestones, securing more investment, and then moving this forward to an end product that's going to be useful. I would say to people out there, whether it's academic science or whether it's industrial science, it's hard, right? And you've got to believe in yourself. You've got to see that kind of goal at the end, and you've just got to accept things aren't going to go away a lot of the time. You just have to pick yourself up, get on with it. And it's a bit like the peer review process, isn't it? Nothing's easy there too. So it's all about determination and believing in yourself and in your team and creating a positive environment for all those people to flourish.
Yeah, that's great words of advice. And I love how you underscore the team. It's not just in entrepreneurial efforts or in commercialization, translation of these therapies. It's really anything in stem cell science nowadays. Gone are the days of the sole operator. It's all about building a strong team and cultivating all these different specialities that you need to reach your endpoint. So I just want to again emphasize that the team, and congratulate you on the teams that you've been able to put together. They're really pushing the needle. Before we let you go, though, we got a couple of science peripheral questions. You're doing a lot of things, Dr. Hay. But the first question here is the thing that you didn't do. What's one hobby that you always wanted to pursue but haven't been able to due to the tremendous demands on your time and effort?
Well, I would say it's probably drawing or painting. I would love to be able to do that. And I'm not so sure if it's a time restriction. I think it's unfortunately my abilities are really limited in this respect. I think my kids from quite a young age could certainly draw and paint much better than I could. But yeah, that's my abilities are limited there.
Well, as someone who similarly has very, very, I would say, weak talent at artistry and drawing, I like to think that science is so artistic, and I really get my jollies out of taking these beautiful micrographs. It's one thing to create, but maybe it's no lesser thing to observe. So I think we're all artists as scientists. Finally, I got a few fill in the blanks for you. First is, when I am not conducting research, I am
going out with my friends, taxi driver to my kids, or working out. Not necessarily in that order.
Well, those are all positive. No vice for this guy. Second, if I could have one superpower, it would be
being able to breathe underwater. I think there's so much of the planet we haven't explored yet, which, yeah, that would be my dream. I'd love to do that.
Aquaman. I love it. I haven't thought of that one. I'm still like with the total banal, infantile wish to fly, but I guess I'll never grow up. Finally, I can't start the day without.
Oh, that one's simple. Tea and coffee. Probably coffee and tea, actually, in that order.
Is that how it goes in the UK? It's not. I thought it was all about tea, but now you're doubling down, huh? The tea plus the coffee. What's that about?
Just gets me through the morning.
Got to get your dose. I found your vice, Dr. Hay, but it's not enough to take you away from saving our livers. I'll tell you what, it's not even noon here during this interview, but I'm going to go have a drink because I'm counting on you to make it all better on the back end. I really appreciate your time and your effort. David, thank you so much for joining us today.
Thank you very much. It's been great to be in the show. Thanks for asking.
All right, that brings us to the end of this episode. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup papers. You can also reach out to us on Twitter at StemCellPodcast or by email at info@stemcellpodcast.com with feedback or to suggest guests. Thanks, you guys, for joining us for this, I would say, bombshell Roundup episode, followed by a really enlightening and fascinating interview. Join us again in a couple of weeks and we'll have something fresh for you. Thanks for listening.